I have the great pleasure of welcoming back a friend of FiRe. I know all of you know who he is. I will do my yeoman-like work at the front, and for those of you who are living on the planet Xeno for the last few years, Craig is the co-founder, chairman, CEO, and co-chief scientific officer of Synthetic Genomics, Inc. He's a biologist most known for his contribution in 2001 of sequencing the first draft human genome, and 2007 for the first complete diploid human genome. In 2010, he and his team announced success in constructing the first synthetic bacterial cell. He's the founder and president of the J. Craig Venter Institute and founder and CEO of the company Synthetic Genomics, Inc. His present work focuses on creating synthetic biological organisms and applications of this work and discovering genetic diversity in the world's oceans. He is a recipient of the 2008 National Medal of Science and is a member of the National Academy of Sciences, and is the author of the book, A Life Decoded, My Genome, My Life. For those of you who were here when Craig last joined us, I still vividly recall the evening, and the part that I remember best is, for most of us, we go to a speech or a dinner thing, and there's probably in that talk, in an hour's worth of chatter, there's probably one or two gems you want to take home. And I distinctly recall in detail the shock of every third or fourth idea or sentence being something that I had to write down in detail and then go investigate later, all of which I thought were world-changing. So, Craig, when I say this, I'm not trying to puff this up. I happen to think that you are probably the smartest living scientist today. And it's a great honor for us to have you with us. Thank you.
Well, I appreciate your compliment. It makes me worried for the rest of the scientific community. So look, it's a real pleasure to be back. I guess I have to come here once every 5 years, so it's but in a way it's a test to see if all the things we threw out 5 years ago, if any of them stuck, and we made some progress, which I think we have. But let me take you through the journey of genomics. Some of this will be review to some of you, and some will be new, but I guarantee there's lots of new. But just to put in context, this is my institute. It's in Rockville, Maryland, also in La Jolla, and then Synthetic Genomics, which is in La Jolla. And then we're on the UCSD campus. We're building the world's first carbon-neutral research building. There's office buildings, but trying to do this with labs and massive computers is a huge challenge. And UCSD has given us the space, so we're halfway between the medical school and Scripps Oceanographic on an incredible site overlooking the ocean. So it's going to be a great place to visit and hopefully to work in. It's an experiment in progress. We're trying to recycle all our own wastewater. If you want to come there for a drink of water sometime. It's not legal yet in San Diego to do that, but eventually, hopefully, it will be. Now, the notion that all the work is based on is a pretty simple one. DNA is an analog molecule with a 4-base system, and when we read that information, we're digitizing biology and converting it into the ones and zeros of the computer. And that's been going on now basically since 1977, when Fred Sanger's team sequenced the FiX174 genome, a small virus. And then it took a lot of time to get to dealing with living forms. So in 1995, my team sequenced the first genome of a living species, and then things really accelerated from there. It was only 5 years after that we did the first draft of the human genome. But on the scale, the first genome we did had 1.8 million letters of genetic code. We had to scale it up to doing the 3 billion letters of the haploid human genome. So the scale of the mathematics, the sequencing, everything else was a huge leap. This is the team at the White House in 2000 when we made the announcement with Tony Blair and President Clinton. The tall guy is my long-term friend, Ham Smith, who turned 80 this last year. He discovered restriction enzymes, the scissors that cut DNA, and he got the Nobel Prize for that in 1978. The guy next to me is Gene Myers, who headed the algorithm development team, and his team wrote over 500,000 lines of computer code in less than 6 months for the new algorithms for assembling the genome. And the guy that looks like Macaulay Culkin is Mark Adams, who led the sequencing team. The labs were these massive, parallel, big machine kind of things, each of these machines, and we had 300 of them at once, were $350,000 machines for breaking the process down and doing it on scale. For computing, in 1999, we had to build the world's third largest civilian computer. It was 1.5 teraflops, which really seems tiny today. We just celebrated the 10th anniversary of the human genome, and this slide absolutely proves that people look better in tuxedos. But it's nice that the whole team's still alive and kicking. That giant facility, it was a 4-story building in Rockville, Maryland, full of machines, now can be reduced to something smaller than this podium. And while it was a big deal for us doing the genome in 9 months for a mere $100 million, that's because the benchmark was the government doing it, in theory, in 15 years for $5 billion. This new machine can do it in a couple hours for $1,000 or $2,000. That's a lot of change in a decade, and it starts to change what we can do. And now, instead of that giant room for 1.5 teraflops, you can get a teraflop accelerator card for your PC. And so, fortunately, it's changing, maybe not fast enough, and it would be an interesting discussion whether computing can keep up with the data demands. In 2007, we actually did the whole genome. We actually have 2 sets of chromosomes. You guys may not know that. One from your mother and one from your father. And what we found is that we differed from each other by a greater amount than we thought. Everybody thought when we only did half the genome, we were only differed by 1 letter out of 1,000 out of the genetic code. And we found on doing, it started with my genome, my parental chromosomes differed by 0.5% from each other. And so we compare any 2 of us, we're comparing 4 sets of chromosomes. So we're somewhere between 1% and 3% different, which is at least slightly more comforting. We have teams headed by Vanessa Hayes at the Institute that are looking at diversity of human populations, and is tracked down by doing the first Bushman genome and then Desmond Tutu's genome, maybe centering in on the oldest human populations centered in southern Africa. And the complication is because we all derive from people that migrated out of Africa, but some migrated back to Africa, and the mixtures are extremely complex. When we look at the DNA comparing my genome to a Bushman genome to a Han Chinese, it has this 1% to 3% range, and it's not unreasonable. When we look just in Africa at the different populations there, there's much more diversity within Africa than comparing Africans to any other population. So there's a new part of getting your genome characterized, and that's what we call haplotype phasing. And it's actually sorting your chromosomes back into what you got from each parent. You know, it's an age-old question of, did I get this trait from my mother or from my father? You know, everybody's asked that question, both about their kids and about their parents. And now we're on the verge of being able to sort that out. And just to show you where the technology is going, Roger Laskin at the Institute developed these single-cell techniques. So we can actually isolate single sperm cells and do a human genome on a single sperm cell. Sperm, hopefully you know, is just half the genome. It's a haploid genome. When you mix it with the egg, that's where you get the other half. So if you do enough sperm cells, you could actually totally segregate the genome into the maternal and paternal chromosomes, because there's about an average of one crossover per chromosome. So if you just do, I would say a handful of sperm, but that has a different connotation, that you can get a really good haploid differentiation. So it's I have to come up with a better term. But in fact, there's these things called compound heterozygotes, where basically variations on the two sets of chromosomes versus what's been done in genetics, not being able to look at that, comes with a much more complex view of what we inherit and how we inherit it. And so people looking in detail at my genome, and it's one of the advantages and disadvantages of having your genome on the internet. People are constantly publishing papers about all the bizarre diseases that you have. And the danger can be, you know, as you saw with the SpaceX launch, we're launching people's ashes into space, but because my genome was the first one sequenced, we've been broadcasting my genome into space for a long time. So if we get an alien invasion, it could be 1,000 copies of me coming back. And that, to some of my colleagues, that's the scariest vision they can have. So it's but looking at these compound heterozygotes, you come up with different sets of definitions. And this is the latest list they gave me of all the new diseases that I'm likely to die from. So I started eliminating the ones of where you die in infancy, because I think I've gotten past that. But so it's going to be a more and more interesting view, but we're at the early stages of this. But even now, it's starting to change the practice of medicine. Pharmaceutical trials have now started to use genomic information. Pharmaceutical companies have fought it for a long time. A great example is Pfizer was doing a clinical trial for a new drug to treat lung cancer, and the trial totally failed until it was discovered by just one scientist by accident, by a mutation in a single gene, all of a sudden makes that drug extremely effective. So over 60% of people that take this drug that have that mutation get rid of their lung cancer. So it starts to become important to know what your genetics are. And this is just early examples, but there's going to be more and more of these. The pharmaceutical industry was worried they wouldn't make enough money off of these. They just found, by checking up the price to really obscene levels for single patients, you know, like $10,000 a month for a drug, just because there's so few patients. But I guess if you're one with that mutation and you survive lung cancer, it works. But I think this will work as the numbers change. Now, you're not alone with just your human genes and 100 trillion cells. You have 200 trillion bacteria in and on your bodies. It doesn't matter how many times you wash your hands, so don't bother. And this is called the microbiome, and we discovered this by just using the same mathematical tools and shotgun sequencing all the microbes in the gut. And in all these different cavities, you have tens of thousands of unique species, and maybe on the order of 10 million additional genes to our mere 22,000. And so a big question now has been, what do these do? What part of human physiology do these affect? And it turns out they affect almost everything. And so it's the newest addition in basic medical science, because we're starting to get all the components together versus everything just being a black box. And so more and more associations are coming up between the environment, your microbiome, your health. And a great example is we just did Larry Smarr's microbiome and found all his intelligence is encapsulated in a few bacteria in his gut. So, so people can pull their ideas out of their asses. I think I should quit after that line. So a lot of associations going on, but it is, as Larry can tell you, it's a lot of data, and it's going to be a huge challenge to sort this out. But yeah, the question, what's the metabolic potential of these 10 million genes? And see that we all just finished a meal. We can take you through this new field of metabolomics, which is using mass spectrometry to measure all the chemicals in your bloodstream. And it turns out, our 22 or 23,000 genes can maybe make 2,500 unique chemicals as part of our metabolism. So if we were to take a blood sample from each of you right now, we'd find about 500 chemicals circulating in your bloodstream. And I would say in California, there's probably a few extra. But only 60% of these are human metabolites. So 30% are from all the fish chemicals and lettuce chemicals and carrot chemicals and wine chemicals. But 10%, or about 50 chemicals circulating through your brain right now, are from bacterial metabolites, of the human metabolites of all the stuff you just ate. And we're just at the earliest stages of learning what these 50 chemicals do. Do they make you feel better? Do they make you feel worse? Do they make you fat? So a lot of challenges, but a lot of new data to start sorting out. So I think that's part of an exciting future. So I think putting all this together, the environment, the host genome, the microbiome, is already starting to be used now by Nestle and General Mills, trying to design new foods based on somebody's microbiome, seeing if you can change diabetes, see if you can change obesity by changing all these components. And I'll talk about it a little bit, but we're actually using all this now to design totally new foods that haven't existed before to try and change these parameters. We'll only start to really make progress when we get some very large numbers, which is why the computing challenge is so great. So we need at least 10,000 human genomes and corresponding 10,000 microbiomes and all the phenotypic information associated with those same individuals. And that will be the beginning of trying to sort out what's nature and nurture. 1 million would help, 10 million would be better. We'll eventually get there. But right now, as I wrote in my book a few years ago, looking at my own genome, it's pretty hard to interpret much because we don't have any real data out there. The genetics community has totally let science down, chasing after single genes and phenomena that are pretty meaningless. So we really need a whole new approach dealing with large data sets and large statistics. It's also now easy to make stem cells. So because my genome was the first sequence, they wanted to look at stem cells. I just gave some skin from a biopsy, and a team with Larry Goldstein and La Jolla worked all the way from making fibroblast in culture to making adult stem cells, then reprogramming these into making virtually any cell in the body. In fact, I went to a restaurant in La Jolla. Those of you know La Jolla, it was 910. There was a very attractive girl that was a hostess there who said, "Oh, my boyfriend's studying your brain cells in culture." You don't get come on lines like that every day. She was wondering what I was thinking. I said, "Why don't you ask him?" But anyway, there's a problem as soon as you take cells out of the body. It turns out this cell-to-cell environment is far more critical than anybody imagined. And as soon as you take these cells out of the body and put them in a dish, and it doesn't matter whether they're embryonic stem cells or skin fibroblast, there's signals that seem to go from the surface to unregulating the genome. So first methylation is lost, and then you start getting massive mutations building up in the genome. So stem cell therapy, as promising as it sounded in the past, only sounded good because nobody was looking at this information. They were looking at the karyotype, which means just seeing if the whole set of chromosomes were there under a microscopic slide, not looking at the genetic code, not looking at all the new oncogene mutations that are cropping up. And sort of as a patient advocate now, I made it clear I didn't want those neurons put back into my brain. And And so the challenge is finding out this new biology of what is there with the cell-to-cell contact that keeps the genome under constant regulation. I mean, that's a huge surprise to everybody. And there's new coatings for plates, putting sugars on the plates, et cetera, that seem to affect it, but nobody's really solved that yet. And we're also trying to see if we can use our new synthetic DNA approaches to reprogram the stem cells to get rid of all the errors. So gene therapy can never work on people because getting the right genes to the right places in 100 trillion cells is virtually impossible. But doing it on individual stem cells or groups of stem cells in vitro is a totally different thing. And people have made genetic changes and they don't induce more changes and they seem to be stable. And so as I start to show you some of the things we're doing with synthetic DNA and the ability to rewrite the genetic code, this could be a whole new era where, in fact, sometime in the future, we will totally make synthetic stem cells and be able to program them for whatever we want. These are some of the challenges going forward. If you weren't upset about all the stuff that you ate and that was circulating through your brain and all the microbes in the mouth of the person sitting next to you coughing, let me tell you about what's in the air. That'll make you feel better. So we started this air genome project, the same thing, just collecting all the cells in the air and sequencing them. And there's was a problem because there's a lot of stuff in the air that interfered with DNA sequencing. And we have these, basically it's the different Dyson. It's a Dyson vacuum cleaner that sucks things out of the air and puts them in solution. And we just filter the microbes and the skin cells, et cetera, and sequence them. We found in New York the interference was iron. And so we just took a magnet, pulled all the iron out of these filtered air products, and you can see it's a pretty massive amount of iron in the air in New York. So it's not clear where it's all from, presumably all the construction and train wheels and things. But all the sampling we did with this was at the level of the 22nd floor in New York City. So we can't see exactly which building it was, but there was a major financial institution in there, which you may understand when you see what was in the air there. So we sampled on the 22nd floor, inside the building and outside the building. We sampled at in and out of a hospital in San Diego, at a home in San Diego, and off the end of Scripps Pier in La Jolla. And what we found was pretty stunning. And this is not measuring free DNA, it's measuring cellular DNA. So the indoor New York air, most of what was in the air was human DNA, human cells. So we slough cells at a pretty massive rate, and when you get a large number of people in a room or a building, that's constantly circulating in the air. But the big surprise was the amount of rodent DNA in the air. And you can see it was not an insignificant percentage. Other animals, bacteria, plants, every office has plants. You go to New York outdoor air, it's almost half is rodent DNA. This is at the level of the 22nd floor in New York City. So if we went down closer to the street, we think this would go up quite a bit. And imagine going into the subway. The big surprise, even though with the financial firm that was there, that it could have been something with the people, but they're out of business, so it's hard to tell. It turns out this was mostly mouse DNA, not rat DNA. The outdoor air was much more rat than mouse, but you can see what's floating around in the air represents that environment. So San Diego is clearly a much cleaner environment. Laguna has to be pretty good too. It's mostly just microbes in the air blowing off of the land or off the sea. The indoor hospital, indoor house look pretty similar at this level. The outdoor of the hospital is more plant and stuff. And go to the endoscripts here, and it's mostly insect DNA, some marine mammals, marine organisms. The interesting thing comes when you compare the hospital to the house in terms of what's there. The hospital's absolutely loaded with virulent microorganisms. I mean, it makes you not even want to go in the place when you see the list of what you're going to breathe in just walking into the building. And so it gives us a totally different way to look at environments. And people have been doing 100-year-old Pasteur experiments of putting out a Petri dish, see what lands on it and grows. DNA is a much better assay for telling us what's there and what's circulating. So it's getting interesting. And because we were talking about the space station, we also have all the HEPA filters from the International Space Station. But just think what happens when somebody goes into the space station. That's a little tiny tin can. So it's somebody with 100 trillion human cells, and they're taking 200 trillion bacteria with them. And each person that goes up is going to take a different set. And it gets pretty ripe up there pretty quickly. So the HEPA filters are like a zoo of life. And so part of what we're talking about for the future for long-term space flight is designing a unique microbiome, sterilizing people, inoculating them all with the same one that will enhance their metabolism and life in long-term flight. So in 1995, we did the first living cell genome. We also did a second one that year of the smallest cell. That was Mycoplasma genitalium. So it has around 500 genes, 482 protein coding genes and 43 RNA genes. And some of you might remember in '96 is when NASA came up with their first of their great claims of Nano Bacteria fossils on Martian meteors. But it was a simple calculation to see that the volume of these was so small they couldn't even have a single DNA or RNA molecule or even a few proteins. And eventually they were declared an artifact. But started a lot of thinking about what minimal life might be, and because we had the first two genomes in history, we could compare one to another and try and make guesses of that. And so we started down a route that has now taken us 15 years to get someplace of asking these fundamental questions about life. How many genes are essential for life? What's the smallest number to define a biological operating system? And this has a lot of practical implications if you're trying to design new operating systems. You want to know what the core is that you can add to in a design sense. And ultimately, after a lot of different failed experiments, we came up with the notion that we actually had to synthesize the DNA and design and construct the genome ourselves, because there weren't the molecular biology tools to get there any other way. So as soon as we started down that route, we had new questions. Would chemistry even allow us to make large DNA molecules? And if we could we boot them up? Or would we just have a very large DNA molecule? So I was talking with George Dyson. I'm a huge fan of his. I think his books are not only the most interesting to read, but extremely educational. And I was joking with him a little bit. He uses life analogies to describe computational things, and I use computational and computer analogies to describe biology. Probably neither is right, but there are a lot of parallels in terms of information things and booting up chromosomes and things that I'll show you. So we started two teams. The first one started on the chemistry. We started by find a way to correct all the errors in DNA. So the machines that synthesize DNA are not very good machines. It's an N-1 situation. The longer you make the piece of DNA, the more errors there are. So we came up with some new ways to correct the errors, and we decided to, for historical purposes, remake the FiX174 genome. So that was the first one that Sanger had sequenced in 1977. And also in 1960, Arthur Kornberg at Stanford discovered DNA polymerase and made a copy of the FiX genome. He had no idea what was in the FiX genome, but showed that he could make this copy enzymatically, stick the copy in E. coli and get virus particles made. So in fact, when we had our announcement a couple years ago, NPR reporter actually pulled out these tapes from Lyndon Johnson talking about how the first synthetic life was made in 1960 with Kornberg injecting this DNA in. So it was more Xerox copy stuff, and they didn't know what they were doing, but it worked. And so we redone that experiment, only starting with digital code in the computer, 4 bottles of chemicals, and writing the genome. Corrected the errors. So then the exciting phase came. We took this synthetic molecule and put it in E. coli. And that's what you see on this. This is a lawn of E. coli on a Petri dish. And wherever there's viruses, it shows up with these clear spots because the virus actually kills the bacteria. So we call this a case where the software builds its own hardware. We just put in this piece of DNA software. It made the proteins. The proteins self-assembled, made this virus. The virus had activity and actually killed the cells as it made billions and billions of copies of itself. So we were pretty happy with this. Our goal was not to make a virus, though. It was to make a large self-replicating chromosome. But we knew if we could make viral-sized pieces accurately, we could find a way to put them together and make the whole thing. So that's what we set out to do, doing this tedious work on the lab bench. And this looks like a Final Four playoff. We would start with viral-sized pieces. So we manufactured these pieces that were 6,000 letters long. And we put 4 of those together to make pieces that were 24,000 letters long. And at this stage, we cloned all these pieces in E. coli, grew up a lot, sequenced it, made sure they were stable, and then went on to the next stage. Put 3 of those together to make 72,000 base pair pieces. Again, stopped and grew them up, sequenced them, made sure everything worked. So this was very slow, tedious work that actually took us a few years. The next stage, then we put 2 of those together and got 144,000 letter synthetic pieces. But E. coli started to object to these big pieces of DNA going in. So we looked around for a new system. We came up with brewer's yeast. Not only did we find it could take very large pieces of DNA, but there's a term of art, it's called homologous recombination. Basically, different genetic systems. Yeast has one. We have our own version of it. That if you have overlapping pieces of DNA of reciprocal code, it can actually cut and splice things and just insert those pieces of DNA. So it's a very powerful system. So we decided we would just put in the 4 synthetic quarter molecules with some designed overlaps into yeast with a vector. And the main thing the vector has is a yeast, chemical-made yeast centromere. So when you look at pictures of chromosomes like a Y, the centromere is the center of the Y. And it's important for replication of chromosomes. So we put in one of those, and we're stunned that yeast just assembled this whole thing for us, and that's how we got the first synthetic chromosome that we reported in 2008. So at the time, this was the largest molecule of defined chemical structure ever made. And we came up with some clever ways or not so clever ways to watermark it, just using the single letter amino acid code, coded for by a triplet of DNA. We wrote the names of all the scientists that had worked on the project, and people thought it was cute but not very imaginative. So hold that thought. So our teams continued to work on improving the synthesis techniques. And so I just told you Ham Smith just turned 80. Clyde Hutchison, who brought the Mycoplasma species to us, retired from the University of North Carolina and joined the team. He's 75. I'm the young guy at 65. But we hired a bunch of young postdocs and things, and one of them, Dan Gibson, looked at all these tedious methods we were doing, and he came up with the method shown in this slide. And it's just a single pot reaction that works at a fixed temperature at 50 degrees centigrade. And all we do is throw in the pieces of DNA if they're designed to overlap properly. And these 3 enzymes that trim back the DNA, stitch it together properly, and made it all just a very simple reaction. And once he got this to work, he his comment was, "I don't know what took you old guys so long." So it always helps to have fresh eyes looking at a problem, but this allows it now to be automated. So we can go from the digital code in the computer to making new chemical code software in an automated fashion. And I'll talk a little bit more about that, because the goal is to get this go all the way through and actually build a self-learning robot that can actually take all the iterations that we have from about 80 million genes our teams have discovered in the oceans, and just do combinatorial genomics, make things, score it, if it lives, you get a check mark. If it doesn't, you find the variations, and you can set up whatever you want to screen for. And in theory, this should be able to learn 10,000 times faster than any scientist can. So I think this could be important if we can eventually get it to work. So we can make DNA now. We can make large pieces of DNA accurately, and that was a huge advance that took us well over a decade. So how do you boot up a chromosome? Well, with the viral DNA, it was pretty easy, because viral DNA sort of infects cells, and that's how it copies itself. Chromosomes, once you get in the bacterial size or larger, are very brittle items. So you can't even pipette them or use them normal lab techniques. So we had to develop new tools to even move chromosomes around. So we have to use electric current to drive them into gels, do reactions in gels, and move them around. But all this led to a real advance in 2007. And I think this is one of the most important papers, at least philosophically, that we've ever published. Because in this study, we basically swapped out the software in a cell, and it completely converted one species to another. All that was changed was the DNA software. Some biologists still object to this because they want there to be more to life, but let me walk you through this, because it's really critical for understanding how all this stuff works. So we have two species of mycoplasma mycoides and capricolum, and they're roughly the same distance apart that we are from mice, on the order of 10% or so. We isolated the genome from M. mycoides, and we treated it harshly with proteinases, because at the beginning of this, we wanted to know if naked DNA could be used to boot up new life forms. If there was going to be a protein that had to be there, it was going to be much more complicated. So we destroyed all the proteins, and then we added a couple of gene cassettes to it so we could select for the chromosome, and it would turn cells bright blue if it got activated. And we worked out ways to move that DNA into these other cells, the process we call genome transplantation, and we inserted it into the capricolum genome. So we have this very cool movie that I'm sure you'll appreciate our sophistication here. We inserted the mycoides chromosome into the capricolum cell. So now we have a species with two sets of genetic instructions. So it's kind of cool, though, what happened. So as soon as we put in the DNA, within fractions of a second, that DNA starts to be red, and proteins produced. And some of the proteins produced early on were the restriction enzymes that Ham Smith initially discovered in the 60s. Those enzymes recognize the chromosome in the cell as foreign DNA and chewed it up. So now we have a body of one species and the genetic software of another. So what happened? In a very short period of time, we had these bright blue cells, and when we interrogated them by sequencing all the proteins, characterizing them, there wasn't a single molecule of the original species in these cells. Everything in the cell derived from the new chromosome that we put in the cell. It dictated all the proteins that were there, that dictated the structure, the chemistry, and as you know, these things go through billions and billions of replications, so there can't be a single molecule left. So this becomes another important concept of the analogy that we're using. Life is a DNA software system. All life that we know is a DNA software system. You change the software, you change the species. They're remarkably simple concepts. but this is coming out of notions that were first noticed basically the year I was born, 1946, where it was shown that DNA was actually probably going to be the genetic material. It took a while to realize it, to the proof that I'm going to show you in a minute. So you might recognize we had a problem. We were actually assembling the bacterial DNA in a eukaryotic cell. So we had to find a way how to get the synthetic DNA out of the eukaryotic cell, out of the yeast cell, to transplant it. So to work this out, we developed a whole new way to clone bacterial genomes in yeast. And it's remarkably simple. We just add this synthetic yeast centromere to the prokaryotic chromosome, and it grows stably as a eukaryotic chromosome. These could be simple step functions in evolution that allow these things to happen at one time. So we can do this. We can grow these chromosomes. We can make modifications. And so we did this with the M. mycoides genome. We grew it in yeast. It was very stable. We isolated it from the yeast cell and did the transplant, and it didn't work. And it took us 2.5 years to solve all the aspects of that problem. And fundamentally, to make a long story short, when we initially isolated the chromosome from the mycoides cells, that DNA had been methylated. So bacterial cells methylate their DNA for different reasons than we do. They methylate it to protect it from restriction enzymes and attack. So their immune system are these enzymes. So if foreign DNA comes into the cell, they can chew it up quickly. Because you can see what can happen now if DNA, just new software comes in, it takes over that species or modifies it greatly. So we isolated all the methylases. We methylated the DNA when we isolated it from yeast, and all of a sudden the transplants worked. And to prove it, we took the restriction enzyme genes out of the capricolum genome from the recipient cell. In that case, we can just take naked DNA out of yeast and the transplants work. So we had this cycle that you see here now. We could just clone a chromosome, make modifications, methylate it if necessary, transplant it, form new species. So at this stage, we were feeling pretty cocky and we're sure that we had now all the components to make this work. And so I decided to scale it up, and we made a genome that was almost twice as large, the 1.1 million base pair genome of M. mycoides. And we did this by making 1,000 letter pieces. We put 10 of those together to make 10,000 letter pieces. We put 10 of those together to make 100,000 letter pieces. And we put the 11 100,000 synthetic pieces into yeast, and it assembled the genome. We did the transplantation, and it didn't work. So software engineers, you guys have debugging software to find the bugs in your software. We had to create a biological version of debugging software to go through and sort out what was wrong with our synthetic chromosome. And it turns out, again, to make a long story short, we found a single base pair deletion in one essential gene. We corrected that one letter out of 1.1 million made the difference between life and no life. We corrected that, redid the transplantation, and we got the first synthetic cells. And so this is what we reported in early 2010. And we call them synthetic cells because everything derives from the synthetic DNA that was in the cell. There's not a single molecule of anything from the parent recipient cell in there. How did we know it was synthetic DNA? And we reported this obviously in the scientific literature, is we put watermarks in because our biggest concern was creating an artifact. A single molecule of contaminating chromosome could fool us into thinking we had made synthetic life. And so we devised a new way to watermark DNA. People thought our single amino acid code wasn't very imaginative. So we, I don't know if you can read this or not, but there's 4 major watermarks. And the first watermark has a new code that allows us to write the entire English language with numbers and punctuation using the 4-letter DNA code. In fact, when we submitted this to Science, we got the review back written in DNA code. It actually drove the Science editor insane because she couldn't sort it out. But it was actually what people had been using for a long time. It was a simple ASCII code, which for coding works okay, but it's not good for biology. So our code puts in very frequent stop codons, so we don't inadvertently create new toxins and new peptides out of just using coded information in the DNA. So we think this code is much better. And when we published this, we just said there was coded information for people to solve. Having being the first species with a computer as a parent, it has a URL in it. And so it actually has instructions. If you solve the code, you were supposed to send an email to the species saying that you'd solved it. And after a number of people that did this, we published the code. And in addition to the names of the 46 scientists and the institutions, we tried to go further. So I came up with 3 quotations, and you'll like one because it's from Richard Feynman, who you were discussing earlier. But the first one's from James Joyce. I don't know if you guys can read those or not. But to live, to err, to fall, to triumph, to recreate life out of life. That seemed like a pretty appropriate quotation. The second is from Oppenheimer's biography, American Prometheus, see things not as they are, but as they might be. And the third from Richard Feynman, what I cannot build, I cannot understand. So after this became public, we started getting a lot of feedback, information. The first thing we got was a letter from James Joyce, estate attorney, complaining that we had not gotten permission to use the quotation. James Joyce was dead. We didn't know how to ask. And fair use laws allow you to use a paragraph without permission in this country. So we dismissed that one. And then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. But if you look on the internet, this is the quotation that you'll find. But to prove his point, this guy sent a picture of Feynman's blackboard with the original quotation, which was, what I cannot create, I do not understand. And I think it's a far better quotation. One of his early biographers made an error with the quotation, so that's what's out on the internet. So just because it's a million times on the internet doesn't make it right. So we've gone back and changed the genetic code so that it has the proper Feynman quote, and he'll rest more peacefully. So why do some of this? I mean, obviously to understand basic life and basic science, but I'm one of the few that believe that science has a role in society, in fact, a critical role. And I've argued recently that we're now at a stage where we're 100% dependent on science for our future. Part of this is because of population. So in 10 or so years, we're going to add another billion people. We'll pass the 8 billion mark. So we're adding 1 billion people in 1/10 the time it took us to double the population of the U.S. And that creates all kinds of problems. And I have a simple equation that is probably not totally a thermodynamic balanced one, but fuel equals water equals food. And it doesn't matter sort of where you start in this equation, they all impact each other. And so we're seeing how and whether our technology can have a disruptive impact. When you look at water, just for each person in this country, it's about 5,000 liters. In the Mideast, it's almost 6,000 liters, even though they have no real sources of water other than by converting oil into water through desalinization. We use a lot of animal feed, and it takes a lot of animal feed plus water just to get a kilogram of beef. I mean, it's just totally non-sustainable as the population is growing. Rice uses a whole lot less, but 15,000 liters to get a kilogram of beef plus all the fertilizer and feed. And in the U.S., we use about 70% of our fresh water for agriculture. agriculture was great for the advancement of our civilization from being hunter-gatherers to having societies, but it hasn't progressed much. And I contend we need to, for all practical purposes, eliminate agriculture as we know it. So we think our ability now to write the genetic code gives us something that philosophers have argued has been the goal of mankind from the beginning, and that's to get control and domination over nature. We've tried it with domestication of animals, we've tried it with agriculture. Now we can sit in front of a computer and in theory, design something. We're also designing new microbial fuel cells that actually convert organic waste material into electricity and clean up water at the same time. And this is what we're using at our institute, but we're building some scalable systems, and we're starting with the most important place on the planet. We're starting with brewery waste so that we can justify making more beer. But it works for almost any organic waste stream, and it also works for desalinization. But I like to think of all these genes that we and others have discovered as design components. The same way that early on the electronics industry had just a handful. And no better history of this than George Dyson's book that I've just finished reading. So we have a lot more design components, a lot more variations, and a lot more that can be done with it. So we actually have software at Synthetic Genomics, computer software for designing biological software, trying to design new species to get them to do what we would like them to do. And we're trying to use the most predominant energy source, and that's sunlight. And we're trying to use CO2 as the fundamental raw material. Instead of looking for ways to bury more of it in the ground, we think we should just recycle it, because everything that happens in biology can start with carbon dioxide as the carbon source. And we're designing new food types, new fuels, new chemicals, et cetera. We're also using these same technologies for vaccines. And so the first genomic vaccines just coming on the market this year in Europe, and it's been a long, slow process. It's taken us 15 years, and it's a new meningitis B vaccine that works against all strains of meningitis B. But what we've been concentrating on with Novartis and the US government is a new way to change how we produce pandemic vaccines, particularly with the flu. So you might remember with H1N1, it took about 9 months to get a new vaccine out there. My institute is one of the major sequencing centers, sequencing flu isolates from around the world. So we're turning it into a computational problem of tracking flus, tracking the genetic changes, trying to predict in advance what's going to happen, and turning around and then synthetically making the vaccines. In fact, we've advanced this now with BARDA and Novartis. Our challenge was to get it under a week, so BARDA now sends us a test pandemic sequence by email, and we have less than 24 hours to produce the new vaccine. So if you saw the movie Contagion, that was really slow crap, man. We can really go fast. So we have it down to under 10 hours for from when we get the email to making the new vaccine stock, which we then get to Novartis for starting to scale up, and they have a multi-billion-dollar plant in North Carolina that uses cells to produce the vaccine, just large amounts of the virus, instead of this 50-year-old technology we still use in this country of using chicken eggs, which creates all kinds of problems for people. So we're trying to advance this. We're basically synthesizing segments of every flu virus ever sequenced, and we have these on a shelf, and we can mix and match very quickly to do things. In fact, our goal is to have the vaccines synthesized and clinically tested each year before WHO even can make a decision what it thinks is the right vaccine for the year, because they don't use modern sequence information, they use 50-year-old technology. So it's changing on that front. But, you know, future foods and medicines. We're actually designing new proteins in the computer that are the most nutritious that you can get. Totally balanced, right amount of amino acids. You can change the amino acid content if you want to increase muscle mass or change where you add muscle mass. So the chemistry, the protein chemistry of food, as you guys were talking about at the beginning, understanding the input is a big part of things, and also just making massive amounts of omega-3s using algae. So we can use these same systems and just design totally new products. My goal is to get rid of, not because I have anything against them, cows and chickens as a source of food. We can completely manufacture beef and chicken in yeast and plants at a scale far beyond with hundreds of times the efficiency of what a cow does in a field, and with much greater food safety and nutrition. So where do we get omega-3s? Mostly from taking dead fish and having naked virgins stomp on them and get the oil out of them. Oh, no, I'm sorry, that's the other one. And obviously this is not sustainable either. And so I just read this interesting article, is the oil you get from algae as good as the oil you get from the fish? Where the hell do they think the fish got it from? The fish eat the algae, and that's where, so they actually should be called algae oils, not fish oils. And the problem is now, those of you who love to eat salmon, most of the salmon you get that's farm raised now is not fed algae with high omega-3s in it. It's fed the equivalent of corn, so you might as well eat steak or a chicken or something because it has no extra nutritional value. So by, in fact, feeding the chickens something different, you can get these high omega-3 eggs, but that can change by a very substantial level. By feeding fish in fish farms high omega-3 algae, we can start to make farm fish healthy again. So in La Jolla, right by Synthetic Genomics, we have a large greenhouse for testing all kinds of new technologies and new algae strains that we're finding and creating. And we just purchased an 81-acre facility in the Southern California desert for scaling up some of these things. Just to finish off, I just want to mention what should come up in questions, I'm sure, and that's the ethical aspects of creating all kinds of new life forms and releasing them on the world. And I was the first to ask these questions and asking for a bioethical review in the 1990s, and Penn State took that on, or University of Pennsylvania took that on and published this report in 1999. Since then, the Sloan Foundation has funded my institute along with MIT and the Center for Strategic and International Studies to do look at security issues, and every branch of the U.S. government has taken part in these studies, and you can download these reports from our website, jcvi.org, if you're interested. And when we announced the first synthetic cell, it was, I think, a scientific first. We heard from the Pope and the President in the same day. President Obama praised the work and asked his new bioethics commission to take it on as their #1 issue. The Vatican said we just changed one of the engines of life, but that it could be very productive for humanity if we can do it safely. And what they told me privately, because they sent somebody around to a lot of my lectures for a long time, they came up and said, we have no trouble with what you're doing as long as you don't do it to people. So there's a narrow definition of who God's creatures really are, but I guess they don't include bacteria, so that's okay. So, I think it's there is an active public dialogue on this. I think what this report from the White House Bioethics Commission stressed, in part because we stressed to them, is making sure there's biological controls being built to in every genome, so things can't live outside of the laboratory, outside of the facility where they're being produced, And a lot of people are now designing new suicide genes and new ways to make sure these organisms are contained. And obviously, there's a lot of potential to do harm with this technology. It's some of the most powerful technology because you only need a few people with some limited tools to make viruses and microorganisms. You don't need armies and billions of dollars. But these are some of the challenges, and I think this is the last slide, just a report from a few years ago from the Royal Academy of Engineering, just saying that this new field of synthetic biology has the potential to be one of the biggest wealth generators for countries, for individuals, for the planet. That has happened in a very long time. Thank you very much.